Pitt Hopkins Research Foundation is committed to directly funding the most promising research in the world to help find a treatment and ultimately a cure for Pitt Hopkins syndrome.
Since 2012, we have funded over $2.5 million in research grants to scientists to support this goal.
We work hard for every single research dollar spent. Our donations come from grassroots efforts of dances, walks, lemonade stands, bake sales, jewelry sales, pretty much you name it, and our families have come together and done it. Our board and officers are parents and professionals who volunteer their time without compensation so that 93% of all donations go directly to finding a cure.
In 6 short years, we have funded transformational research. Our research strategy is focused on creating repositories for our cells and models, increasing our understanding of the basic science of TCF4, along with evaluating potential treatments and possible cures with protein replacement and gene therapy. This multi-pronged research strategy will greatly increase our chance for successes. An equally important part of our mission is that the Pitt Hopkins Research Foundation recruits the best scientists and physicians who share in our mission.
Hi, I'm David Sweatt. I'm the chairman of the pharmacology department at Vanderbilt University. I started doing research on Pitt Hopkins syndrome because AUDREY DAVIDOW sent me an email out of the blue and asked me to start working on Pitt Hopkins syndrome. Our approach began with using gene engineering to try to create a mouse model, a laboratory mouse model of Pitt Hopkins syndrome, and see whether knocking out the Pitt Hopkins gene in a mouse gave us behavioral differences in the mice that looked like they might reflect the differences in Pitt Hopkins patients. One of the most exciting things about this work is it does look like, indeed, these mice are a viable model for trying to understand Pitt Hopkins syndrome, and most importantly, to be able to use them as a drug screening platform to try to identify new therapies for Pitt Hopkins syndrome.
Hey y'all, my name is Niswender and I work at the Vanderbilt Center for Neuroscience Drug Discovery in the Department of Pharmacology at Vanderbilt University. So in lay terms, what we're trying to do is to develop compounds here that eventually would be ready to go into a clinical trial with time in order to begin exploring hypotheses clinically. Can we impact a clinical indication instead of curing a mouse, for example? And so one of the things I'm really excited about is that one of our targets, actually several of our targets, look like they could have regulatory sites for TCF4, which is the protein that's mutated in Pitt Hopkins. And so from a basic science angle, I'm really excited about exploring the interaction between TCF4 and some of the targets that we're working on here at VCNDD.
Hi, my name's Annah. I work in the Sweatt and Niswender laboratories at Vanderbilt University. My work is focused on validating a new target for Pitt Hopkins syndrome. What really inspires me in working on Pitt Hopkins is the wealth of knowledge that we've acquired in the past few years on TCF4 and the pathology of Pitt Hopkins syndrome. I think there's been an explosion of knowledge in the field in terms of what we know and new avenues that are being researched, and I find that really exciting.
I'm Ben Philpot, and I'm the Associate Director of the UNC Neuroscience Center.
Hello, my name is H.M. Lee. I'm a research associate at Philpot's lab.
I'm Sally Kim. I'm a grad student in the Philpot lab.
So we're working on several different projects in the lab. One is a gene therapy approach to try to treat Pitt Hopkins syndrome. We're doing this in collaboration with Steve Gray's lab at UT Southwestern. We're also working on an approach to activate TCF4. And we're also looking at NMDA receptor hyperfunction in Pitt Hopkins syndrome.
My major job function is drug screening.
So my study focuses on understanding the spatial temporal expression patterns of TCF4 and its function in the brain.
I think what's great about research is we're now at a point where we're beginning to see progress. We're really beginning to understand the pathophysiology of Pitt Hopkins syndrome. We also have tools both in terms of having mouse models that are appropriate to study and in terms of having appropriate viral vectors to use for gene therapy. So we have a lot of tools at our disposal, and we see that progress is really being made.
Hello, my name is Alysson Muotri, and I'm talking from my lab here at University of California in San Diego. This is what we call induced pluripotent stem cells, where we reprogram cells from the body of the individual into this pluripotent state. And from that state, we can recreate what we call a mini brain or a brain organoid. As a consequence, when we make these mini brains, it's an avatar, it's a clone of that individual that we have in the lab, and it should work or it should give us mechanistic insight into how the brain of that specific person works. Most importantly, when we make those mini brains, we don't make just one from each person, we make thousands of them. And the fact that they are tiny structures allow us to test drugs or new medicine, hoping that one can fix, revert, ameliorate the condition by testing thousands of drugs at the same time, instead of a couple of them or one by one. So it really speeds up what we call drug discovery. And also importantly, because it has a structure of the human brain represented in there, we can test other therapeutic opportunities such as gene therapy. So we can see how the vectors will deliver the correct gene or will edit the genome to fix the correct mutation into a more realistic system that mimics the human brain.
My name is Brady Maher. The focus of my lab is to use animal and human-induced pluripotent stem cells to model Pitt Hopkins syndrome. We use these model systems to identify physiological differences between controls and Pitt Hopkins samples. We then try to understand the cause of these differences downstream of the TCF4 gene with hopes of identifying a suitable therapeutic target. I am most excited about our work identifying a sodium channel that appears to be upregulated in Pitt Hopkins syndrome. We think that blocking this channel with a drug could provide therapeutic benefit for Pitt Hopkins patients. In addition, we have new data from a large collaboration between several Pitt Hopkins Research Foundation labs that indicates mutations in TCF4 results in deficits in myelination, a process that insulates neurons in the brain. We think this new data opens up novel avenues for treatments for Pitt Hopkins syndrome.
My name is Guilliams. I'm the co-founder and CEO of Healx. Healx is a technology company from Cambridge University focused on predicting treatments for rare and orphan diseases. Together with the Pitt Hopkins Research Foundation, we focus on drug repurposing. So that's looking at existing treatments that are already available, safe, and understand whether some of those could be used for Pitt Hopkins. It's the quickest, safest, and cheapest way of finding a potential treatment for a disease. And at Healx, we focus on using machine learning and artificial intelligence to do so effectively.
I understand how difficult their situation is and that the lab is really working hard to try to improve things.
I would like the other parents to know that I'm also a father of someone with autism, that there are more researchers out there like me who wants to speed it up, find opportunities for a translational type of application.
It's moving at a great pace right now. I know it's not moving ever fast enough for the parents, but as scientists we feel like things are really starting to pick up and we're really making positive progress.
We've seen our children at war with their body every day, not being able to communicate their needs and wants, some not walking, most not talking, and many with seizures. We believe that one day it will be scientifically possible to make their lives better. That time can't be soon enough. We believe in a cure. Although we've come so far, we still have a long way to go. To accomplish this, we need a village, our village, to help fund research and to support each other on this journey. I want to make everyone here a promise that the Pitt Hopkins Research Foundation will not stop working until we have that cure. I want to make you the promise that our work and our dollars will be spent boldly and wisely. This is urgent. We are not doing this for the next generation. We are doing this for our generation of kids. Please join us in making Pitt Hopkins history.
I have an opportunity to make a difference. And I think that's really what I want to do and what my lab wants to do, is to try our best to make a difference and to improve someone's life. I'm Ben Philpot, and I want to make Pitt Hopkins history.
We want to make Pitt Hopkins history.
I want to make Pitt Hopkins history.
We want to make Pitt Hopkins history.